This is the beginning of DNA synthesis. First, the topoisomerase comes through the double helix, relieving the stress before the DNA strands reach the enzyme helicase. Then, the enzyme helicase comes through and unwinds the double helix, forming the leading and lagging strand. Single-stranded binding proteins attach to the DNA, preventing the DNA strands from coming back together. Primase forms primers, which start the formation of an Okazaki fragment. The lagging strand forms Okazaki fragments because it synthesizes in the 3 to 5 prime direction. This happens because DNA polymerase can only synthesize in the 5 to 3 prime direction. Okazaki fragments are formed by a primer first coming in and laying down a few complementary nucleotides. Then, DNA polymerase takes the place of the primer and finishes forming the Okazaki fragments. After the Okazaki fragments are formed, an enzyme, ligase, comes through binding the Okazaki fragments together, forming a daughter strand. Now onto the leading strand. Synthesis in the leading strand is easier than the lagging strand because it runs in the 5 to 3 prime direction, which is the only direction DNA polymerase can synthesize. To start the synthesis of the leading strand, a primer is laid down to start the pairing of the matching nucleotides. Then, the primer is replaced by DNA polymerase, which finishes the synthesis, forming a daughter strand. Now, DNA synthesis is complete. The product of the lagging and leading strand are semi-conservative strands, which means they each contain old parent strands and a new daughter strand.